Okay, and we're back. Let's do a quick review question. So, which of the following is not a major innovation that the sponges first evolved? So pause your computer. This one has a lot to read. And so the answer to this one is E. So the sponges basically invent a new way of reproducing with sperm. Yes, we've seen sperm before, but it's a different kind of sperm in the animals. That's technically the synapomorphy is the unique animal-like sperm. Um, they are multicellular with specialized cells that allow them to feed in the way that animals feed. And then they also have a very simple but functional skeleton that allows them to be much larger and support their body more than typical protists can do. The thing they don't have are tissues. There we need the metazoans. Okay, so let's keep talking about tissues. So we already know from much earlier and throughout the semester that when it comes to animals, there are four kinds of tissues: muscle tissue, nervous tissue, epithelial tissue, connective tissue, that are grown from muscle cells and nerve cells and epithelial cells and connective cells. Again, this is our structural hierarchy. All animals have these except for the sponges. So if you're a metazoan, you've got these four tissues. Metazoans can move, they can feel, they can react. Sponges can't. Now let's go back to our structural hierarchy and think about you have cells, you have tissues, you have organs. So how do we build organs? It's one of the great stories in really all biology, but especially zoology. And the way that organs are grown in animals is embryologically during development in specialized groups of layers of cells called germ layers. And here, germ doesn't mean like a microbial pathogen, it means to grow, to germinate, to sprout. So, this is a growth layer within the embryo. And so you can think of it this way. Here's your zygote, the single-celled diploid thing after fertilization, and then it becomes the embryo. And there's a lot of detail here that you do not need to know. If you study embryology, you'll learn all of this in detail, but it divides 2 cells, 4 cells, 16 cells, keeps going. Eventually, it becomes a hollow ball of cells. And then it starts a process of what's called invagination, or puckering in. The cells start to push into the hollow chamber. Okay. And at this point, we have a set of cells on the inside and a set of cells on the outside. And these at this point are called germ layers. Now, in humans, this happens pretty quickly within a couple hours to days. But once the cells are germ layers, then the cells become special identities. And so basically, there are three possible germ layers in animal embryos. There's a ectoderm, which literally means outer skin. Dermatology is a skin doctor. Ecto means skin, so or outer. So this is an outer layer. And so in this picture here, that's the blue set of cells. They're not blue, we just color code them. There is the innermost or the endoderm, the inner skin, which is the endoderm in yellow in here. And then in some animals, there will be another germ layer that forms in between these called a mesoderm or middle skin, mesoderm or a mesoderm. And so once you're at this germ layer stage, then the cells start to take on different identities. And so the cells of the ectoderm are going to develop into the organs of the skin and nervous system. It includes the sense organs, feeling, hearing, smelling, tasting, the brain, the nerves. This is an outer part of your body thing. And although these things are inside your body too, they're mostly on the outside. You don't have sense organs inside your body. The mesoderm is going to be rich in muscle and bone and connective tissue and blood and basically the part that is inside the skin. And then the endoderm is the gut. And although it's weird to think of this, a gut is really a layer of skin functioning and feeding that's a hollow tube that goes from your mouth to your anus and basically is inside your body. Okay? And so all of the digestive organs of the gut, the intestines, the stomach, the esophagus, all of those organs, the liver, pancreas, they're grown from the endoderm. So the organs that work together in the same organ system develop from the same layers. It's a really beautiful process of embryology. And to be honest, plants do something just as cool, but I'm a zoologist, so for me, this is just really awesome. Okay, plants are pretty developmentally awesome too. So if you were to take one of these embryos, when it has a germ layer, and cut it in half, there's two versions. One embryo has the hollow ball of cells and then a germ layer lining it called the endoderm, and then nothing in the middle. It's basically gelatinous, and then an ectoderm on the outside. And we call these animals diploblasts, meaning double layers. Blast is a word that you normally think of exploding, but it really means kind of coming together, so growing. So double layers or double growths, diploblasts, and they have an ectoderm and an endoderm. And if you have these germ layers, when you develop into an adult, you will be radially symmetrical. There's some other things they have as well. For example, they have a mouth, but no anus. Okay, if you have all three, an ectoderm on the outside, an endoderm that's going to line the gut on the inside, and then a third germ layer between these called the mesoderm, then you're a triploblast, meaning three layers. And then when you're an adult, you will probably be bilaterally symmetrical. So the embryology, even very, very early in the embryological development, is going to determine what the adult basically is going to be organized like. It's a really cool aspect of development and embryos determining adult conditions. And so here is a jellyfish that we've cut in half. If you did this when it was alive, it would feel it because they have nerves. And we've color coded the gut, which is lined with the endoderm and basically builds the stomach. We have the blue, which is the ectoderm derived stuff, which is mostly skin. And if they have eyes, they're going to be there and feeling and organs and stuff like that. And then the red in between is just a gelatinous area. You don't need to know the name. So jellyfish are gelatinous because they don't have much muscle. There is a little bit of muscle. They have some muscle in their skin. They have a little muscle in their gut, but they don't have a lot of muscles. And so they don't move very well. If we were in person, I would do a dance, pretending I was a sea anemone or a jellyfish to show you pretty much they can't do much. In contrast, bilaterians have muscle. They've got just as complicated skin in a lot of cases, derived from the ectoderm. They have as complicated a gut, but now they have an opening at one end, and follow it through, there's going to be an anus at the other end. And then they're going to have a lot more blood in the middle and muscle, and later on, maybe lungs, and hearts and circulatory system. And there they will be able to move much more. They will be able to pump blood greater distances. They will potentially be able to be bigger, although they don't have to be. But you're a much more sophisticated animal if you are a triploblast, and you're going to be radially symmetrical. So let's add this to our phylogeny. And again, in the textbook, they don't put these traits. Trust me, we're building a better phylogeny. So here's our metazoans. They have tissues and organs. Now we can add ectoderm and endoderm, which means that the groups that branch off are going to be diploblasts. Then we have the evolution of the mesoderm, which technically means triploblasty, and then bilateral symmetry. So these things, the germ layers, really are the thing that drive whether you're radially symmetrical because you only have two and you're diploblastic, or you gain the mesoderm and are now going to be bilaterally symmetric.